keep saying the 4th edition one, but we're in chapter 18 for 5th edition, 16 for 4th edition. Okay, so this is kind of some cool stuff, not just because it's exciting to think about when things go wrong, but it's actually really impressive how well our cells are able to deal with errors in our genome. Okay? And then the other piece is this is all, so this concept of mutations and recombination, right, which are both topics we'll be covering in more detail. This is how we get evolution, right? So this is like our raw material for natural selection to occur. We have to have variation, which often can come through random spontaneous mutations and or recombination, right? So this is how evolution happens through natural selection. So beside the being excited about it morbidly, because like mutants are cool, this is how we can get variation by having these mutations happen. Now, of course, some of them are bad. Okay, they're deleterious to our cells and to the whole organism. So we've also evolved to have cellular mechanisms to help repair them and fix them, to make things right again, hopefully. So, but to a certain extent, we will see a certain percentage of these mutations carry from generation to generation. And we're going to kind of start this topic thinking, I'll try to describe some of the types that you guys are responsible for. There's tons and tons of types of mutations. But we're going to go through the types of them. So it's kind of more of a definition-y concept section. And then we'll go into some of the causes, how these things happen. Okay? So you can run out there and avoid smoking and tanning beds and pretty much everything in the world. Just hide in a dark place. Okay, no, don't do that. So we're going to go through types first. And we're really only focusing on gene mutations, okay? So when we start our discussion about types of mutants or types of mutations, we're just considering those that are in coding regions, okay? So so we're not really, for this course, there's actually some really neat mutations that happen to enhancers and silencers and the non-coding regions that regulate these genes, that turn the on-off switches, right, that turn these genes on and off. If you mutate that area, you also have an issue and have a problem. But for this class, we're not going into that. So we're really focusing on genes themselves. What happens when we disrupt the normal gene sequence, okay? And from there, focusing on gene mutations, we're going to look at two types, like two main kind of categories, and then I'll list a bunch of subcategories. The first one is those mutations that change the single nucleotide one base pair. Okay? Somehow they're affecting only one base pair. And then the other category is we're going to look at it's a kind of a form of copy number variation, but it's when you see a mutation that affects the numbers of copies of a repeated sequence. So if you have like a 2 or 3 base pair repeated sequence, say you have GCC, 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 over and over and over again for 10 times, that's repeated, and that's normal. Whereas if you have 5 times or you have 450 times, that's when we start to see abnormal mutant effects, okay? So this falls under a category, a type of mutation. It's mutations that alter the repeated sequences. And they're small repeated sequences, not big, long stretches of repeats. Small in the sense of the repeat itself is 2 to 3 base pairs repeated over and over. Okay. And you can actually put, for that section, we're sort of going through a case study. So in your notes, if we get to that today, which we might not, this is number 2, it's called Fragile X. That's our case study we're going to go through. Or our example of a disease that's caused by a mutation that alters the numbers of copies of this repeated sequence. Okay. So our first big area is this number 1. So mutations that affect single base pairs, okay? And they're another name for them, instead of saying mutations that affect single base pairs or one nucleotide, they're called point mutations. Okay? So referring to the fact that it's at one point on that DNA sequence. And I keep using the word mutation, which is correct. But keep in mind, again, that this could be beneficial or not. This could be a pro or a con for the organism, it just depends. Or it could be neutral. So we'll walk through those different possibilities where we can see that at a protein level, what that might affect. Types of point mutations, you can have a base substitution. Okay, so that means you're exchanging one type of base that was originally in the normal sequence, and you're exchanging it out with a different base. And there are 2 sort of flavors or types of base substitutions. You could either change, so you could change your base from a purine to another purine, which is a slightly less of a big change, right? So if you just move from a purine nucleotide and whatever the mutation cause was, the result was another purine. Okay. So that would be, let's see, so this, and same, conversely, I should add, this is the same as if you're talking about pyrimidine to a pyrimidine change. So these are your A's back and forth to a G, or C back and forth to a D. So if C was in your normal DNA sequence, somehow it gets changed to a thymine. Or if thymine was your normal sequence, somehow it gets changed to a cytosine. So this type of base substitution is referred to as a transition. You're transitioning, but you're not actually changing from a purine to a pyrimidine. So the alternate type of base substitution is when you make that exchange. You go from purine to pyrimidine, or vice versa. Okay. That's referred to as a transversion. So if you're doing a transversion, or if that's what occurred in your sequence, okay, that's when you have purine being converted to pyrimidine, or vice versa. So your C gets changed to an A or a G, etc. Chemically, this makes a difference to actually separate those out into 2 separate categories. To call these transitions, to call these other ones transversions, because you're making a bigger chemical difference, and especially if you're thinking about something that was once a purine, or sorry, once a pyrimidine, and now you're plugging in a purine, the bulkiness factor of that lesion in the DNA makes a difference. If you're going from a 1-ring structure base to a 2-ring structure base, all of a sudden you're bending the whole backbone of the DNA, you're making a bigger change. So we differentiate between whether it's a transition or a transversion. Okay. This is kind of just defining stuff right now. Okay, the other thing you can do besides substitute bases is you can actually flat delete one or add in a new base. So people in the genetics world who think we're cool, we call these indels, just to sound like we know some special fancy language that no one else knows. But really that just stands for an insertion or a deletion. Okay. But if you want to sound ultra cool and walk around and be like, yeah, we were talking about indels the other way, and no one will know, and you will be dirty and be still people. So for insertions, okay, or deletions. People really talk like this. Like I walk around in the hallways in graduate school and people are like, oh, I was looking at the indels and my sequence and the CPG islands are just totally crazy in that area and blah, blah, blah. And you're like, I'm thinking about manatees and fun times. Okay. Indels. Insertions or deletions. I'm not going to draw anything else with that. You're either inserting a base or you're deleting a base, okay? And it can be any base. So we don't break it down into a transition transversion thing. You can be sticking in any of the 4 bases and at any place along the gene sequence, or you could be deleting one. That's what that basic category means. What we are going to think about, though, is functionally, what does this mean? So what does this mean for the protein? All these different, this idea of a base substitution or an indel. Okay, if you're doing any of these mutations, how does that affect amino acid sequence? Okay, so how does that affect your codon reading sequences? So there's different consequences of this. So this is at a more functional protein level. And in your notes, it's kind of like consequences for results area. So this is at the protein level. I often wouldn't realize how nerdy I was becoming until I went outside of my bubble and would have dinner with normal people, and they're around your graduate student fellow people, and they're all looking at them like, you guys are straight nuts. This is not cool and fun, and But you guys are all headed on that path. Aren't you excited to see what all going on? Not necessarily graduate school, but anywhere else, anything you do after this, whether it's jobs, professional school, graduate school, you guys will all get together and talk all nerdy about science. Because you must like science if you're sitting in this room. To some level, you may hate genetics, but you like science. Or you just like manatees. Okay, so what happens to our protein? Is it possible nothing happens?
Can't you get a frame shift? Does your frame shift?
Yeah, so one of the options would be a frame shift. Okay, so one of the things that would have a consequence. So I'd ask, is it possible that nothing happens? But yes, it's possible something happens. And one of his ideas. So this is a frame shift. Okay. And what that means, we often with genetics we refer to in the coding sequence, we call it the reading frame. Okay, and that it's just like you would think of if you took text out of a book, if you took a sentence out of a book and you deleted one of the letters and then crammed the letters all together based on the organization that you were originally reading. So if you had to read in these 3-letter chunks, which is how your ribosome is reading off the codons, right? So you could have, I don't know, AGG, ACT. Okay, but this is your normal wild type. What we mean with a frame shift, so what has to happen for a frame shift to occur? No one's saying indels. I'm supposed to say indel and be cool. Nobody learns anything. Indel, right? You insert or delete something. Yeah, so you guys are correct. You insert or delete a base pair and it throws off the sets of 3 codons, okay? So if you deleted, I have an example somewhere. Or say we stuck in, you insert a G, right? If you insert a G there, does that disrupt our first codon? No, not if we start reading from this left-hand end. We're reading left to right, just like you would, in a textbook or magazine or whatever. So your AAG stays the same, okay? And all of a sudden we have an A, an inserted G, a C, and this gets shifted down so that now you're reading CTC, and then we have the rest of the whatever else is hanging off the end of the protein. But essentially, you've changed your amino acids. You've actually changed the whole reading frame. So your ribosome will go along, chug along, and keep producing amino acids, a whole polymerizing your protein, but it's going to look potentially like a really different protein from the one you originally started with, right? And you potentially that could cause a stop or some other problem in the reading frame. And same with the deletion. If we took out one of these, depending on where we take it out, and that should also, in terms conceptually, if you have an indel, all fancy like, early on in your amino acid sequence versus late, how might that change the consequences of the protein? Would you prefer it to be early or later on in the sequence? Later on in the sequence, yeah. So later on in the sequence means you have more normal amino acids being produced at the beginning. So you might have an almost completely normal protein except for maybe the very end.
You didn't say that. You added an
Yeah. On a second. TCCT.
Okay. Aha. No, yes. Ish.
Yes.
Eh. There. See, I was thinking about meanties instead of. Okay. Frame shift makes sense? Okay. So what else? What else can we have happen? Some of this is kind of, should maybe be somewhat review from a silent, right? So that was kind of my original thinking about if you made a substitution or transition transversion that somehow doesn't change the amino acid sequence. So that you might also see this called synonymous. Okay, so silent mutation is the same as a synonymous. So that means you have the same amino acid as the result. Okay, and there's a ton of examples where you could go from arginine and change it to a C, for example, and you still have arginine. Okay. So that would be what? Transversion or transition? From an A to a C. Yeah, transversion. So you had a transversion, which is a base substitution. Kept your amino acid the same though, so luckily, maybe your protein's completely fine, it's silent. We don't see a phenotypic, we often don't see a phenotypic outcome, there's no observable phenotype from a synonymous mutation. What else? Synonymous, frame shift. Another idea. It makes sense, yeah. So, missense mutation, which really means what? We've created a different amino acid instead of the same. And you'll also see this called non-synonymous. In addition to missense. So this would be, we could take glycine and change it to arginine. So now we have a whole completely different amino acid. We changed from an A to a G, so it's a type of transition, right? Not a transversion. Because you went purine to purine. The thing about missense is this can have varying degrees of a consequence too, okay? So in terms of what happens to the protein, this can have a variable effect on the protein. From a minimal effect to a really big effect. I mean, you're only talking about one amino acid difference, which doesn't seem so terrible, but it also depends a little bit on the chemistry, right? If you're going from lysine to arginine, you should kind of think about too, I'm not gonna like make you guys draw me the structure of amino acids on your exam or something, but you remember back somewhat to your terribleness of memorizing those, right, that some are more basic, some are more acidic, some are more polar, some are non-polar. So if you think about the chemical consequences there, you could have some pretty big ones, especially where in your protein sequence this happens. Okay? So if that amino acid substitution happened in the active site of a protein, you might have some pretty big issues in terms of the function of that protein being wild type or normal. Okay? So thinking about where this effect happens and kind of the type, what amino acid change it is exactly. But we refer to that as a missense. We have one more? One more.
Nonsense.
Yeah, nonsense. The best kind. Okay, so nonsense means you made a base substitution that has created, or you've done a frame shift, essentially, which could be an endo, right, insertion or deletion, that has caused there to be a stop codon where there normally was not a stop codon. Okay, every protein, every amino acid sequence has a stop codon at the end, but now all of a sudden we're putting one in where it didn't have one. As an example. Let's say you change this front C to a U. Now all of a sudden you have your amber stop codon here at the end where you started out with glutamine, something like that. Might be. Might have to check another one. To amber stop. And again, if this happens, kind of like the frame shift, if your nonsense mutation happens very early on in the protein, you could potentially have a super truncated protein, which is a way bigger issue than if you maybe just lopped off the last amino acid, and now you're just missing, now instead of 20 amino acids long, your protein's 19, you might have some level of function, depending on the protein. Okay. A lot of this was review, yeah. Okay. Okay. So causes, this is the, or at least to me, this is the cool stuff. Okay, so we've defined some different types of mutations. We're going to get back to that repeated sequence one. So we've looked just at the point mutations right now. And we'll come back around to the Fragile X example of increasing or decreasing repeated sequences. Now we're going to think about some different causes, ways that these mutations happen. So, as you might imagine, some just happen randomly, okay, and there's nothing you can do to prevent or stop them from happening. And then others are sort of induced or externally caused, okay? So some of these, we're going to break them down, and I'll let you know when they're more of a spontaneous type, which would be more of an internal cause, like cellular internal, or they're induced in some external way. And I don't mean purely experimentally, but this can happen due to environmental external conditions attacking cells and things like UV and other carcinogens and things like that. Okay. So our first category here, so the spontaneous ones. What are some ideas? How might these happen spontaneously? Where do we see errors?
DNA.
Okay, in the DNA, but what's the cause?
Lack of checkpoint.
Yeah, lack of a checkpoint, but what kind of my questions too about what kind of cellular mechanisms would we expect to see an error happen? At what point? So that's talking about checkpoints, which is our cell cycle. So somewhat during methylation, but what point in the cell cycle? G phase, S phase, N phase? Jason? Yeah, S phase. Yeah, a big point when we go to replicate. Okay, so DNA polymerase itself, remember, is error prone. It can make mistakes. Okay, it's not a perfect system every time. So the polymerase itself during replication, you can have errors. This enzyme's not perfect, and it can certainly insert the wrong base. And it's often because of something called tautomers that we'll come back to. So these are variations on the chemical variants of the regular bases that exist in our cells. So you can kind of like star that, like, yeah, I'll come back to this. She threw a word up on the board, and we don't really know what it means. We don't care right now because I'm in zombie note writing mode. So tautomers are something we'll come back to. They're a variant of the base. Okay, it's like having bases that look almost like thymine, but there's a random different functional group hanging off the side. And so polymerase gets confused by those and can sometimes stick those in, or it encounters one of those and doesn't know what to do and puts the wrong base opposite it. So we'll kind of come back to that. But also, inherently, this enzyme's error prone. Okay, so it makes mistakes at replication. And keep in mind, we're going to be talking about how the cell repairs these. So most of the time, the cell can go along and repair it. Okay? We're going to get to repair mechanisms definitely next week, not this week. What's another spontaneous? I mentioned one actually earlier with cellular metabolic byproducts. I did mention that earlier, though. I did say carbohydrates earlier. Yes, they're listening. Antioxidant. Yeah, think about antioxidants, radicals. Jason's doing the wave. Yeah. Yeah. So the reactive oxygen species. So this is these metabolic byproducts can damage your DNA. So this is your byproducts are. This is your regular cellular processes going along doing their thing, and some of the products that come off of these chemical reactions are harmful to your DNA. It just happens that they're around. An example is what Jason had just mentioned, which is the reactive oxygen species. So hydrogen peroxide is one. You have that superoxide radical, which I think has like a dot.
Is it just O2?
Is there a dot? I don't know. This is crystal. I can't remember the nomenclature for that. And I think the hydroxide one is OH with this is a hydroxyl radical, O2 superoxide. I think it's a negative charge. Yeah. And it has a dot to show your extra electron thing. Okay, so clearly not a chemist. So these are, think of them as like high energy. They're very destructive in your cell. Okay, and what they tend to do is they tend to add on oxygen groups to your bases. Okay, they tend to change the chemical structure of the bases, which then of course causes problems with other reactions. Okay, so it's called oxidative damage to your DNA. Changes the chemical structure by adding in hydroxyl groups or oxygens. And typically to the ring structure of the nitrogenous base. So not so much, they don't affect the backbone or the sugar, the phosphates or anything quite as much. It's more to the base itself. What happens so after you have these hanging around in your cell and changing the chemical structure of your bases, what often happens is then they get mispaired. Okay, so that's kind of the subsequent result in terms of creating a base substitution. So this results in mispairing. And then what happens is that replication, so you'll see a lot of these mutations, especially the ones that, where I'm gonna get into depurination and deamination, where we're changing the chemical structure of the base, which means the base is still in there, it just looks a little different, right? Usually where you see the problem is then subsequently when you go to replicate your DNA, the polymerases don't really know what that is, and they don't know how to handle it, and they don't know how to make a complementary base, right? Because normally we split it apart, we have our template, and polymerase can be like, you're a G, so I stick a C here, you're an A, so I stick a T here, you're a sleepy T, so I put an A here, right? But then you get to here, and you're like, that's weird, it has an extra oxygen hanging off the end of it, I don't really know what to do with that. And so that's when we see the problem. Okay, so kind of with a lot of these chemical mutations, changes to the bases, it's more that you see the transitions, transversions at replication. Sounds like duck goose. Okay. So it's when we get to replicating our DNA that then there's the cell has more problems. Okay, so the next two again have,
Well, my question is, if DNA replication is occurring and it's in helices very broken up into two strands, can the reactive oxygen species interact with bases on the template strand, or is it just nucleotides being added on?
they can certainly react. You mean, can they react when they're hydrogen bound to their pair in a double helix?
Interact with the backbone, like the DNA bases on the backbone, and the reactive oxygen species interact with those, or the nucleotides being
So I'm going to rephrase your question to make sure I understand it. But so when DNA is in a double helix structure, has not single stranded, when it's in a double helix, can the reactive oxygen?
Well, it's when it's broken up by helices, so when that double helix is broken.
Yeah. No, they definitely can react with that. I might be misunderstanding, so you can come talk to me later. Yeah. No, but you certainly can, whether it's in the double helix form or whether they've been split apart by helicase. Yeah. Typically, you might see more react with any of these, like the deamination, depurination, any of these chemicals. So number 2, 3, and 4 on our list are all sort of like chemical modifications to the bases. And those tend to be, as you can imagine, or they're going to occur more likely if the DNA is out. So if the chromatin's all packaged in around the histones, you have less area for a reaction to occur, less atom surface space to be attacked on by another. So yeah, typically it is going to be more at replication, at transcription, when you're unraveling your DNA so you can transcribe it, things like that. Yeah, that's when they're more prone to attack. Anyway, third one has to do with purines. Okay. And it's when you actually remove that covalent bond to the backbone on a purine. So it's called depurination. I'll write a little definition up here so you have it in your notes, but it's really what we're talking about here is removing a base, essentially, and the way that you're removing it is with that covalent bond between the actual sugar, the deoxyribose, and the base gets broken. And the base we're referring to is a purine. That's why it's referred to as depurination. So your A's and G's. And the reason that it is broken is this particular bond is somewhat unstable. Okay, so this, and this is where you guys don't need to be experts in the biochemistry behind this. Certainly not for Bio 311, but just know that in your head, between a purine versus a pyrimidine and the sugar, this bond is a little bit more unstable. And so what happens is, H2O can come in, so water, which is everywhere around in your cells, right? Available in there, can have a reaction. So water can come in and make an attack and it releases the base from the sugar. The overall outcome here is we lose a base and we lose a purine. Okay? But what this means in terms of instability is H2O our base gets detached. And again, the cell can repair this. Okay, so we have our so here's your double helix going along. And if you I feel like I have to use red when I'm doing mutations, so I don't know why. So if we took a purine and it gets depurinated, so through chemical processes you guys don't need to know, big black box, H2O can come in and react with it, release it from the sugar. Okay. And your cell can repair this, but again, if it comes to replication, depending on what point the cell is in replication, if these strands are going to, just as Jason and I were just talking about with helicase, they're going to be separated. You're going to have T, G, C, A, just doing fine along the bottom. The top, if you still have a gap, okay, let's be giant mark. Okay, if that doesn't get filled in or replaced, if that base doesn't get replaced, polymerase has to make a decision here about what base to put in. Okay, when it's making its template copy, right, it's going along and putting in the correct bases to copy, and then it's getting up to this upper strand, and it doesn't know which base to pair up with for the blank spot. Okay. And we'll get into kind of what polymerase will do, how it'll try to handle that. But that's, again, where we start to see problems is more at replication. And one of the ways, and we'll get to that repair mechanism, but one of the ways it can fix this is by looking at this other strand that's available, right? So those repair proteins that then come in, grab this other strand out, and are like, hey, you idiot, there's a C there that clearly wants a G, stick a G in. Okay, so they actually tell each other, they talk. See, sometimes I'm sarcastic and you guys are just such blank faces, and I'm like, oh, they're actually writing in their notes, hey, you idiot, put it in. I told my 242 class that neurotransmitters are always purple, and no one blinked, and they just wrote purple, I think. So, there's that. So replication, we see issues, okay?